animals. And we will continue with Kaya and Dagmara. They will give us a lecture first and then practical session in the laboratory. So we will try to... Não ouvi outra coisa que posso apresentar. Ou assustar, né? Ok. Just give me a minute, please. Okay. We will try with presentation. Hopefully it will work. It might take some time because Kaya and Dagmara have video.
Yeah, but don't worry about the video. It's not.
Okay, we will try. So
It's not necessary.
we have just enough time to give it a try because you put so much effort to present it to us. We can at least try to present it. Okay. So here it is. Share. So this. Well, give it a time. Let's try. Here it is. And now like this. Just put the slideshow on.
No, we don't have it, Ivana.
I know. I will try one more time. Just please.
Yeah. If it's better for you, no, that's working. I mean, if it's better, you can also stay on the not full screen, just minimize then the sidebar and the upper menu bar that we can have a better, but like this, it's perfect. Thank you.
Perfect. Okay, but it's not working. Oh, it's working. Okay, perfect. That's what I learned.
So I'm going to start, if you don't mind. Hi everyone, I'm Dagmara, this is Kayo. We are microbial ecologists based at Biology Center of the Czech Academy of Sciences in Budweis in South Bohemia. I'm going to sort of continue with Silke's topic. She was focused on the very targeted isolation of obligate parasites of specific algae. We are trying to decipher the broader community, which includes parasites of algae, which are facultative, not necessarily obligate. So this is our take on how to try to find them and get them into culture in natural environments. So we have titled the talk Chaos Fungorum. Probably know this is a term that was coined by Carl Linné in the 18th century because he was really frustrated with all these algae that could not be fit into the neat little boxes, right, of the systematics. And we are still frustrated in the 21st century, I think. But it's almost like, you know, the last frontier in the aquatic system. It's what makes the research really exciting. So we're just learning as we go. I will try to not keep you too long with the theory so we can go and play in the lab later. This is the outline of the presentation. So I'm just briefly going to go over the current attempts at classifying these guys. So there's zoosporic organisms, both in the true fungi section and also in all the other more or less related clades and groups. So I will not talk about chytrids because Silke already did that really well. So I'm going to focus on some of the other perhaps less known groups which contain zoosporic parasites of algae. And then trying to look at the different freshwater niches which can harbor these organisms, which would be probably good to focus on, trying to get them into culture, just to resolve the really complex life cycles, get more sequences into databases so that we can maybe design probes for better isolation under the microscope. Looking at some ecosystem boundary processes that could also influence the presence and activity of these organisms in the aquatic environments. Silke already talked about the seasonal importance or importance of season in planning your sampling and isolation, also grazing, and some of the approaches that have worked for us so far. So there's multiple attempts. Let's start with the true fungi and the basal groups there. There's basically two current systems of classification. I think the first one, the Tedersel one, is the one that is used more broadly. Not all of these groups contain organisms that produce zoospores. I have put the blue zoospores next to the groups that do have them. Also, not all of these groups will contain parasites of algae. For example, this group are anaerobic fungi that are fermenting cellulose in the guts of mammals and animals. And as far as I know, they have not been found outside in the environment so far. So Silke has already explained what zoospores are, what their morphology is. So I'm not going to go into that. I'm not going to talk about chytrids. Chytrids are probably the most well-described group out of these guys so far. Another group which is important as parasites of algae is the Aphylidium mycota. These guys are actually obligate, most likely. So far, I think 4 genera have been described, but I'm sure more are coming. So their main trophic stage is a plasmodium, which phagocytizes the host cytoplasm and then digests it internally inside of the big digestive vacuole. The big vacuole is probably one of the distinguishing morphological features, although as you can see, it's the morphology is extremely difficult to follow under a microscope. Here the arrows are zoospores. You can see that these are not the classical uniflagellate zoospores that you saw the chytrids have, but can kind of have these pseudopodia, amoeba-like movements and so on. So Aphylidium mycota are one of the groups. Then there's Blastocladium mycota. They're pretty common. They are also uniflagellated at the pole of the cell, like in the chytrids. Their hosts are pretty broad, so they not only parasitize algae, but also invertebrates, plants, other fungi, oomycetes, and so on. And I think Gabriel talked about Paraphysoderma in the morning. That's a really nasty one, right?
Yeah.
So you can see it here parasitizing the Haematococcus culture. What I'd like to show here is this amazing plasticity in zoospore morphology. So you can see that the zoospores can go from the typical flagellated, fast-swimming zoospore up to amoeba-like, slow-moving, and it's still, it's the same species, right? So the morphology is extremely plastic here and extremely difficult to identify anything. Another basal group, the Roselomycota, it has been thought that these guys are only parasitizing on other fungal hosts or oomycetes, but there's been a paper which described the Roselomycota parasite on Coleochaetes. So probably algae with a more complex structure, branching algae, things like Cladophora and stuff like that can also be infected by Roselomycota. So also a single flagellum, polar flagellum. You can see here the little black dots are the cysts attaching to a host hypha. Here you can see the sporangium forming inside of the hypha and some resting spores inside of the hypha as well. One of the morphological features that is pretty distinct is kind of spikes on the resting spores. So this designates these Rosella species. And here at the bottom, you can see a cloud of zoospores being released from the sporangium. Then we're going on to fungi-like organisms, the oomycetes, which are hyphal. So they parasitize larger algae, filamentous algae, they're intracellular. They have zoospores with two flagella. One is a bare whiplash flagellum, the other has tinsels or hairs on the flagellum. So that they resemble hyphae or form mycelia similar to what true fungi would look like, but they rarely have septa. So this is how you can tell them apart. Sometimes they have septa on the reproductive structures. Right, so this is the famous video, which is not even ours. It's from YouTube and it's not playing. Okay. Well, I just wanted to show you how fast these zoospores are, but I could also just give you the YouTube link. I wanted to show you the important thing is the chemotaxis and all mycete zoospores and all zoospores, right? They orient themselves based on chemical cues in the environment. And I think this probably could maybe in the future be used to bait them or to attract them into some kind of traps. So yeah.
Maybe try to push the right button.
With the right button? No, don't worry. Yeah. Oomycetes are also fascinating because they can actually go from, growing a sporangium, maturing the zoospores and releasing the zoospores in like bare minutes. So that's one of the fastest developmental processes in biology. It's amazing. Which tells you how intermittent this is if you're looking outside in the environment in a lake or a stream or a river, and it's extremely difficult to catch these events, to isolate. Another group of the fungi-like parasites are the labyrinthulomycetes, also very interesting. A lot of them are saprobes. Some of them are single-celled with these three kind of spike-like pseudopodia, I guess you could call them, and some of them form sort of ectoplasmic mesh or a net over a substrate, and use that for as sort of a highway for the cells to move over the substrate. And these are also extremely diverse, many different groups and clades. I have put these orange arrows into the groups where algal parasites were identified. Another one, Perkinsozoa. These are obligate, also parasites, mostly marine, of dinoflagellates. which are characteristic, some of the species, by forming these beaded sporocytes, which then divide to release each of these divides to form two individual zoospores. So this is also quite typical for the group. And another one, Syndinialis, these are actually early or basal dinoflagellates, which are infecting multiple hosts, multiple different hosts. So again, intracellular parasites, you can see the trophon stage inside of an algal cell. This is the healthy one. This is what it looks like as the infection proceeds, and the digestion of the host cytoplasm. Right, and so you can see the morphology. It's really hard to distinguish from all the others, like chytrids, anything else just under the microscope. Cercozoa, also very common in freshwaters, extremely diverse morphologically, ecologically. You can find amoebae that are either naked, they can have sort of cases or testae made of protein. They can be flagellated, they can have pseudopodia, or they can have both. So this is total chaos. So here is a species of Gromia growing on a diatom. And Phytomyxia, these are parasites of plants, higher plants, but also can be found parasitizing on higher algae, which have more complex thalli bodies. Right, and so all of these guys will be present in the environment. So here's a picture of a water body, and just like idealized processes that go on there, which involve fungi, not only parasites, but also saprotrophs, their grazers, their hosts. There's huge heterogeneity, temporal, of course, seasonal, but also horizontal, also vertical, especially in deep water bodies. So you have different processes happening in the photic zone and the euphotic zone and the littoral zone at the level of the sediment. And there's also the connection to the terrestrial environment. And there are, cross ecosystem boundary processes that we don't know about, but probably some or a significant part of the inoculum of these groups can come or originate in the soil. So algae are not just present in aquatic environments. There's a lot of soil algae that thrive whenever the soil is moist. These will also be infected by parasites, and the whole system is then transported into a recipient water body when there's, for example, a rainfall. So you get movement of the water from the soil, from the watershed into the water body. Then that will create the biotic interactions and so on that we know absolutely nothing about so far. So this is the terrestrial source of the inoculum. Some of the examples of the sources is, of course, plant litter is a very good source. So the leaves come into the water body already colonized, and are very good substrate for parasitic species that are not obligate biotrophs, but have a saprotrophic part of the life cycle, and can be a reservoir of these taxa in a water body. Same goes for mineral particles that are associated with organic matter colonized by soil microbiome, which contains both bacteria and different fungi, different resting stages, spores, which also get flushed into the aquatic environment and can represent the source of the inoculum as well. So when we look into the actual water body, what are the different niches, except for the obvious plankton, right, phytoplankton, where we can find these guys and try to isolate. So we found that a really good source material is fecal pellets of grazers, especially things like copepod snails or algivorous fish, which, Silke showed the plankton net. So these guys kind of act as concentrators. So as they graze on the phytoplankton, they concentrate not so much zoospores because they digest the zoospores, but different resting stages. And so the fecal pellets, if you collect them, can be a source material for isolation. Organic matter flux, definitely. So, this is a marine snow in the marine environments, but same organic matter, also sediments in freshwater environments, and that is heavily colonized by all sorts of organisms, bacteria, fungi, protists, and so on and so forth. Egg masses, I'm missing a picture here. So basically, gelatinous colonies, egg masses formed out of carbohydrates, like things like amphibian eggs, insect eggs, or ciliate colonies, or even bryozoan colonies. So these are not only, they're colonized by algae, okay, but they're kind of like a garden colonized by all sorts of things and can represent a good source of inocula also. And then if you have a water body with macrophytes, with conditions, light conditions that are good enough and the morphology of the water body allows, then macrophytes are excellent sources for inocula for these parasites there. As our macroalgae, actually. So Cladophora, we did some comparative studies and found that the microbiome of Cladophora is actually similarly diverse as that of the macrophytes. Also gardens that contain a lot of things are good potential niche to isolate from. There's periphyton growing on them, but there's also periphyton growing on, or epilithon growing on rocks, and so on and so forth. So also a good source material to isolate. Oops, sorry. Right, so this is just to illustrate the diversity of the niches and the heterogeneity of the environment, and the amount of biotic and interactions within there that will all affect the isolation success that you need to take in account before you go out and isolate. So it really helps to know the water body that you're sampling prior, to do some maybe pilot sampling to get to know the system. So you know what you have there, where to look and when, where and when. So this is just, we looked at our sequencing data that we did, and we tried to look for these basal fungi groups if we can find them, and we found them basically in every single sample that we had, pretty common in pond samples, also in hemiparasitic plants and their hosts, and as endophytes growing in plant leaves, also in cave sediments. So of course, not all of these are parasites. Most of them would be saprotrophs, right, heterotrophs, but the potential for facultative parasites being there is very high. Grazing, another factor. I think both Gabriel and Silke already talked about grazing. So just briefly, zoospores are extremely advantageous to eat because they have very good stoichiometry. They're super rich in lipids, polyunsaturated fatty acids. So a whole spectrum of grazers will readily eat zoospores in aquatic systems. So this is a picture from one of our traps of amoebas. That there was a sample stained by DAPI, which stains as a fluorescent stain, which stains nucleic acids. And so you can see the amoeba, two different species of amoeba. This is the nucleus here, this big blob. And the amoeba is just packed full of zoospores that it's been grazing on. So Grazers are a really important factor which structures and influences the dynamics of infection in aquatic systems. Seasonality, Silke also talked about this. Seasonality will have different patterns depending on the trophy of your system. So in highly eutrophic systems will differ a lot from oligotrophic ones. So you can see that here, this bar here is parasitism. If you look at the gray curve, that is your phytoplankton biomass. So every system is different. So it's important to get familiar with what seasonality actually occurs in your system, and then try to pick the best time for sampling based on these metadata or other data. This is something that is just started to get attention from researchers, but you know that in basically all higher organisms, humans, for example, the colonizing microbiome has a great impact on the outcome of infection, right? So it can mitigate or allow infection, and so on and so forth. So obviously a single-celled alga will not have a very complex microbiome, but if you go to higher algae or even to algal colonies, these are colonized by a pretty diverse spectrum of mainly bacteria, producing secondary metabolites, and these can also play a role in the outcome of infection within an algal population. But the research is still at the very beginning. Right, and I think I'll give the floor to Kyle because he has green fingers and can isolate like magic.
So now I talk a bit how the main approach that we are using in our lab to isolate fungi. And since when you have an idea and how to collect the samples and how to bring the samples to the lab and how to proceed with the isolation. And of course, the first thing, you need to have some question that you want to get some answer. And after you have your question, of course, you go to looking for the place that fits better to give you some reply. Here, my focus will be in the environmental samples. I will not focus on algal production or like this, because this is the purpose of our projects. But of course, it can be adapted for different other issues. It's be possible to do it. So of course, the first thing, you need to select your place of study. And when you select your place, it's better to have any information about it. So all the maps, how to read the place, to look the surround of the aquatic system, for example, or look for some papers that you can have more information about previous studies realized on that place. So here I'm just showing the place that we collected the samples that we are playing today in the lab. So it's a pond in south of Český Budějovice in Czech Republic. So basically we collected subsamples in these big ponds that you can see here. And also there is a stream linked of these ponds that we also have some samples. And nearby this stream, we found a very small pond that was basically part of the water that was outflowing the big pond. I will show some pictures later how it looks like. Maybe I can show now. So this is the photo of the area that we collected the samples. So this place is the big pond that we left there some fungal traps, and we also collect the water. Here, this yellow arrow is showing the small pond that is fed by the water from this big pond. And here in the last part, we can see the stream that I show in the map. And pay attention that these three different environments, they are very close to each other. And you will see later how they can differ on the samples that you collected that you will see in the microscope. And here's me, so just to see scale reference. And of course, when you are going to the fields to collect some samples, you need to know if you need permissions or not. And this is just basic things. And you also need to pay attention in also in every safety guidelines, how to get there, how to avoid any problems in the field, because this can be a big problem. Have a team to go there and never go alone, because this can be a problem. But I think this is just obvious things when you go for sampling the field. Also, it's always a good idea to have a checklist, all the equipments and materials you need to have when you go to the field. Because if you go to the field and when you get there and you just realize that you forgot essential equipment or essential material, you need to go back if it's possible, and or you just lose some sample or miss this, and this is terrible when you plan this in advance, or we need to get the sample on that. So basically GPS, rubber boots, protection for the pants, boats if you need to collect the water in the lake or some big pond, samplers for water, nets, and also bottles. And think if you need some sterile equipment there, like the bottles, if you need to collect some samples of water to look for microbial community later, it's better to be sterile before. Some probes to get the information about the environmental parameters like pH, temperature, oxygen, and some thermal box to transport the samples to the lab and then proceed with all the experiment or methods. Basically, when you are planning to work with microbes in general, not only fungi, you have two different approaches. They are not excluded. You can use both to be complementary, but you have one approach that you need to cultivate the microorganisms. So it's a cultivation dependent method. So sorry, it's wrong here. So this is all the methods that you need to cultivate the microorganisms to study later, or you have some methods that you don't need to cultivate the microorganisms. So you don't need the isolation. So basically you have the samples, it can be soil, water, other kind of sample. If you are looking for independent cultivation methods, basically you play with the samples directly. You can, for example, extract the DNA and use some fragment to look for sequencing and look for the composition of that sample, or you can look for some fingerprint methods that you compare the sample, the composition, or even use some markers or stain to look in the fluorescent microscope to identify which kind of microorganisms you have in that sample. So basically, you don't need to cultivate the microbes here. On the other hand, these methods, you need to isolate the microorganisms and then continue with your research. We have two different methods. Today, in the lab, we will look for the serial dilution, but we have also the per plate. But basically, you take your sample and then you dilute the sample in many fractions. And this dilution basically is just to have some, when you have a sample with high concentration of cells, you diluted it. So in the less dilutions, basically you can get access to more hairy microorganisms or some microbes that take longer to grow from the plate. And after the serial dilution, we just inoculate a liquid of the sample on the plate. We spread this liquid on the plate to have a more homogeneous growth in the medium. And then we go to incubate the samples in appropriate temperature. And after some time of incubation, you can see which microorganisms grow on the medium, on the surface. The only difference of the pure plate is that you inoculate the serial dilution directly in the medium, and in the still liquid media. So it's before the media solidify, and then you pour the media on the plate, and then you can see the growth on the media later. But in this method, you can also isolate some anaerobic or facultative anaerobic microorganisms, and this is only aerobic. So just keep a focus on the serial dilution that we will demonstrate later in the laboratory. So we collect some samples. These samples are not diluted, of course. So you make some dilutions. Usually we work with 4 or 5 dilutions. It's the ideal to get some difficult microbes to isolate. We inoculate part of these dilutions on the plate. Usually I inoculate 100 microliters. And then I spread this inoculum on the surface of the medium, and I incubate this on the temperature. So the important thing of this method is to select an appropriate medium. So you need to think which kind of fungi you want to isolate. So according to this group, you select the proper medium to grow this. Also, the temperature, when you are working with environmental samples, it's important to know which temperature that microorganism was used to live on the field. Because if you want to select the fungi was present on that time, you need to offer the same conditions of the environment. And so not only temperature, but also pH is a very important parameter that we need to consider. Usually fungi likes to grow in pH between 3.5 and 5.5, 6. They're like more acid than bacteria, for example. The incubation time, as you talked before, usually 7 days, you can already see some growth on the plate, but some fungi need more than up to 30 days, 40 days. So usually I keep the plates on the incubator for 30, 40 days, and then to check every 2 or 3 days if I can see some growth or not. And to avoid the other microbes when you're looking for fungi, I used to add some antibiotics. So usually streptomycin, tetracycline, or chloramphenicol. And the concentration of these antibiotics depends of your group that you are working, but usually the range is from 100 micrograms to 300 micrograms per liter. And you can also add some other inhibitors to avoid the growth of fast growth fungi, like ascomycetes. When you're not looking for this group, you can add some inhibitors like rose bengal that you can see these groups growing on the plate, but this group will not grow so fast. So basically, you will not have an overgrowth of these fungi, and then you can also select the fungi that grows not so fast, that they are more slowly. So the medium that we are using in our project when you are looking for zoosporic fungi is basically these 3 medium. They are more properly used for chytrids, all of them. And these PMTG, they are also used for blastoplasma mycota. Unfortunately, we still don't have any medium for the other groups that Dagmara was talking before, for the other zoospore fungi, because to develop a new media is also a big issue when you are working with isolation. So you need to think about which nutrients you need to add in the media, what other compounds, the elements that the fungi needs to grow. So basically, this is the most common media that people use it for the isolation. And today we work with this PMTG that is the most range used for chytrids, blastoplasma mycota, and recently we are very successful to use to isolate oomycetes using this medium. And the other part of the practice today we are talking about fungal traps. It's a method that we are using to collect the fungi. And this method is based on baiting methods. So the baiting method is a very common method that is used for many groups that works with chytrids. So basically the baiting method is you go to the field or you go to your culture of algae, you collect your sample, you bring your sample to the lab, and then you can add some baits on the surface of the sample. And after like 7 days is usually ideal for start to observe. You can see the growth of some chytrids or other zoosporic fungi on these baits. These methods, the fungal traps, basically we put the baits on the field. So we developed these traps, these chambers, these slide frames. So we use this to leave the baits on the field. We use a nylon net to put the baits inside into this net. So the net is basically just holding the baits inside. And we leave this trap on the field for like 5-7 days, or even if you want to keep it long, it's possible. And one advantage of this method is that you don't need to collect the sample in the field. You bring the sample to the lab, so you can just go to the field and fix the traps there, and time to time you can go to collect it if you want to see some seasonality dynamic, or you can just come back after 7 days and just collect and bring to the lab, and then proceed with the isolation of the fungi. And which kind of bait we can use on these fungal traps? Well, this will depend of the group that we are looking for. So if you are looking for groups that are more related to cellulose degradation, you can focus on some baits that you can find cellulose. Or if you are looking for some groups that they are more able to use chitin, so go for baits that use chitin. So the baits is what you want to attract. So this is what you define your decision. The main baits that we use today is cellophane because of the cellulose. This is very easy material to find and very easy to work. And also when you are looking the microscope later, you see how it looks like. So you can see the cells more clear. Also, we are using onion skin. So the white onion skin that's very thin. This is also easy to find, to get it in the lab. It's easy to sterilize. All these baits we sterilize before to add in the environment. So you can be sure that the fungal that you get there is from the sample you're working. Also, we can use snake skin for chitins, or some pollen are very common material to use also to attract fungi for these traps. Corner husks that is also easy to work, or also even some exuvies from insects, crustaceans, or arachnids. Or you can also just collect these exuvias from the place that you are going to work, and then use as a bait. Well, we are also working with some sediment traps in our project. So because one of the aims of our project is to look for how the fungi can be transported from the terrestrial environment to aquatic environment. So basically these traps, we have 4 different traps that we are working. We have this trap that we set them on the inflow of the aquatic system. So basically we can collect all the particles that is coming from the streams that surround. And we have also 3 other different traps that collects all the particles that just sit from the water column to the sediment. We have another pictures here. So this is the trap that is in the inflow of the aquatic system. And here is the structure that is holding the other 3 different traps. To say something? It's because this traps is very new in our project, so we are still beginning. So what is the next? When you have the traps and then you wait for the incubation time in the field or another system that you want to looking for, you need to go there, take the traps and bring to the labs these traps. And we usually analyze the traps, part of the baits in the microscope to see what is there, what is colonizing that bait, and then part of the bait we use to inoculate in a plate to isolate the fungi that is there. So basically, we transfer part of the bait to a plate. So we'll do this in the lab. So with aid of tweezer, you can just screen the plate with this part of the bait. Of course, we usually work in a flow laminar box to avoid any contamination from external area. Today, we will not do it because we will be in the lab, but usually keep this in mind that it's very common. And after some time, you can see the growth of the fungi in the plate. And when you see the really start growth of mycelium, usually you need to transfer this mycelium to a new plate to then avoid contamination with the other fungi growth around, and also to have sure, or at least to try to get a poor culture in your new plate. So for this, it's better to check the plate every 2-3 days. Then when you see in the binocular or in some other equipment that you can check the plate properly, if you see some mycelium or any hyphae start to grow, it's better to transfer this fungi to a new plate, because if you leave there and the other fungi starts to grow, you'll be completely mess, and then you cannot isolate the fungi more. So after the transfer, usually you incubate this plate in upside down in the incubator to avoid the contamination by water. As these fungi, they can produce zoospores. Water can be a very easy way to contaminate the other plates. And will be the next steps is to storage and keep the maintenance of these colonies. So basically, you can use nutrient agar slants when you want to store this for a short time of period, or also Castellan methods that when you store these plugs of medium with mycelium in Eppendorf tubes with saline solution, for example. This is a method for medium time, like 6 months to 1 year. It's the spare time for this method. Or you can also store the cultures for a long term using, for example, cryopreservation or also glycerol, and storage this in -8 freezer. Other step that you can go after that is for identification. I think Silk talked very well about this. So you can go for morphological traits, you can go for DNA extraction, select some general primer for this group, and then do the sequencing. And very important part is to deposit this sequence when you have later or any identification in database, because we need to improve our database. We have a lot of gaps in the database, mainly for zoospore fungi. So the two more important that I would say is NCBI and Microbank, that other people can use later to compare, to identify your cultures. Or you can also send the strain for some culture collection to deposit your strain there. And we have one collection now in US that is very specific for zoospore fungi, Cesar, but we have others culture collection that is very common for not only fungi, but also bacteria and algae, as ITCC and TSMZ. Well, today in the lab, we work with the traps we brought with us So the traps are from that pond that I showed you, the string and the small pond. We will see how the traps looks like in the microscope. We have 4 inverted microscopes here in the lab. So for that, we will use the lactophenol blue to stain the samples. It's not a very specific stain for zoosporic fungi, but this can stain chitin, so it will help us to see something. Usually, it's better when you have more than one method to stain the sample to identify the group that we are looking for. But basically, what we see is something like these pictures we got last week. So we can see like a very entangled hyphae on the baits. We can see also some sporangiums full of spores or empty. It's better to, this stain is very tricky because the lactophenol blue stain chitin. So for example, some algae groups, you can also have this compound. So for example, here we can see that cluster of algae. So it's very important to observe and see if you can see some other characteristics, not only the staining cells, that you can say that if it's fungi or not. We can see some chytrids, of course. Also some algae cells with parasites, as you can see here and also here. And some hyphomycetes. So I hope we can have very fun in the lab to looking for these dates. And there in the lab, I will show you how to prepare these fungal traps. I brought some for you to practice a bit. We use cellophane to prepare these baits. And we also go for some cereal dilution. So I will just demonstrate to you how I do the cereal dilution and how I plate this. And then we go for the baits to look in the microscope and stain this with lactophenol blue.
Perfect. Thank you.